Hello. Today I will be talking to you about the lactobacilli or probiotic bacteria and their role in the gastrointestinal tract. Probiotic bacteria are microorganisms found in dairy and fermented products, such as yogurt, and are essential to host digestion. They are known to fight off pathogens as well as maintain or even strengthen the immune system. Although prior to their benefits, they must make a treacherous journey through the GI tract, passing through the stomach and small intestine, which contain high acidity, before they reach and are able to reside in the large intestine. Here is a sketch of a person consuming some yogurt that contains lactobacilli. Under the microscope, the bacteria are rod-shaped and considered gram-positive. Once the yogurt is ingested and chewed, it is swallowed and follows the esophagus all the way down to the stomach region. This is the area that gets a little tricky for the probiotics to be successful due to the high acidity. The stomach has a pH level of 2, which is actually quite similar to that of a lime. And due to the high acidity, probiotics seem to decline in numbers. But in some cases, the bacteria does have a tolerance towards low pH and therefore have the ability to continue their journey. After the food is digested in the stomach, it makes its way into the small intestine, where the pH level goes up to about a 4, which can be compared to the pH level of cherries. And finally, as the yogurt passes through the small intestine, the digested material containing the probiotic will enter the large intestine, in which it will have the opportunity to benefit host digestion. Now that I have taken you through the journey of the lactobacilli in the gastrointestinal tract, I can show you the way that we simulate it in the lab. We began by culturing the probiotic from a liquid supplement that can be found over the counter. The supplement then can be directly spread onto a plate that has MRS agar, or nutrients that help the bacteria grow. And then the plate is placed into an incubator, preferably one that is 37 degrees Celsius and contains a percentage of CO2. Then you should wait a couple of days to let the bacteria grow. Once a bacteria is grown, we can do a growth curve to simulate the GI tract. Similarly to what happens when a person ingests food, start by inoculating the mouth control and mouth treatment tubes. And after 10 minutes, transfer 110 microliters into the corresponding stomach control and treatment tubes that has 3 replicates. Do the same transfer after an hour into the small intestine tubes, and again after 3 hours into the large intestine tubes. While you wait for the allotted time to transfer media into the next stage of the tract, you can carry out 5 serial dilutions for each tube, which just means that you are diluting the concentration of bacteria, making it a little easier to count colonies in the next few days. After you finish the serial dilutions, you can begin spreading plates. An example of spreading plates is pipetting 100 microliters from the second dilution and transferring it into a plate that contains MRS agar and spread the liquid around the plate. Once you finish spreading the plates, place them into the same incubator as you did with the original lactobacillus and wait a couple of days. After 48 hours, you should be able to count the colonies. A helpful technique to count is make grids along the plate and count each grid and then add up the total number. Finally, when you are done counting, it is time to calculate CFUs over milliliters, or number of colonies times dilution factor over volume plated in milliliters, and then graph the results. Hope this video gave you an insight of the way probiotics work and how we are able to test their growth in the lab setting.